Well, hello to everybody. Welcome everybody to the 9th session of the GO2NE Global Ocean Oxygen Network webinar series. We are very happy that you join us today and our incredible speakers this morning, afternoon, or evening in several parts of the world. So to begin, let me start with some housekeeping. You as attendee have the possibility to type your questions in the question box. You can also use the chat box to share additional information or if you have any logistical problems. But please try to use the question box to submit your questions. If you want to speak at one of the upcoming webinars, please submit a short abstract. We will take that into consideration, and the link will be shown in the chat box. But now let us start. We have two great experts speaking today. They both are from Peru. We will start with Dante Espinoza presenting for 20 minutes to be followed by 10 minutes of questions and answers. And afterwards, Renato Salvatteci talk again for 20 minutes and 10 minutes of questions and answers. So now let me introduce you Dr. Dante Espinoza. Dante is a researcher of the Marine Institute of Peru, IMARPE, in the Laboratory of Oceanographic Modeling, Ecosystem and Climate Change. His topic research is focused in the ensocycle and the impacts of the climate change using biogeochemical models. Dante holds a bachelor's degree in biology and a master's degree in marine science from Peruvian universities. And in collaboration with IRD, he achieved a PhD degree at the Sorbonne University from Paris, France. The PhD thesis was titled The Interannual and Decadal Variability of the Primary Productivity and Oxygen Minimum Zone in the Peruvian Upwelling System, which afterwards led to some recent publications. Today, Dante will give us a presentation entitled Numerical Modeling of Oxygen Variability and the Oxygenation Trends in the Southeastern Pacific. So now, after these words, I pass the floor to Dante. Dante, you are welcome to start your presentation.
Okay, thank you. I will try to share my screen. Do you see my presentation?
Yes.
Okay, nice. So, well, good afternoon. Well, firstly, I want to say thank you for this opportunity to give this talk. My talk is about numerical modeling of oxygen variability and deoxygenation trends in the Southeastern Pacific. Well, this is a work in collaboration with IRD. I work in IMARPE and specifically the laboratory LOCEAN, no? With Vincent Echevan and François Colas. Well, if you see this map shows the concentration of oxygen in the subsurface water. Red color means low concentration of oxygen. You can see here that the region of the Humboldt current present one of the most intense and shallowest oxygen minimum zone in the open ocean. And also these volume waters are important because are related, for example, with the decadal variation of some species like anchovy and sardine from Peru. So the OMC limits the habitat of several species. Also, in the last decade, we observed that, and several papers also demonstrate that the oxygen minimum zone are expansion in the last decade. For example, this is a paper from Strama 2008, where we can see that the white line limit the oxygen minimum zone, and we can see that in the last decade we have an expansion of this zone. Well, but what are the factors that influence in the formation and the maintenance of the oxygen zone of Peru. We can talk about, for example, remote forcing and local forcing. About the remote forcing, we can mention, for example, the equatorial undercurrent that brings oxygenated waters to the in front of Peru. And also we can talk about two branches of the equatorial undercurrent, for example, the primary and secondary jets that also influence and shape the oxygen minimum zone. Like local forcing, we can mention, for example, the winds that influence in the upwelling, no? And that welling brings low oxygen concentration of water, no, from deeper layers. And also we can mention, for example, mesoscale activity and the Kelvin wave that we will see later what is the influence of these processes. But also Peru is impacted by El Enso cycle, no? For example, during El Niño event, this is a profile in front of Peru in Callao, no? In red, in orange and red means that we have oxygenated water. And in this box, we can see here that we have oxygenation during, for example, El Niño 97-98. But during La Niña, we have the opposite effect, no? You can see here, for example, the oxygen minimum zone. This is a vertical section of Peru, also in Callao. And you can see that the oxygen minimum zone is shallower during La Niña. So we have less oxygen during La Niña condition. And regarding to the trends about oxygen, there are a couple of papers that show, for example, time series of the oxycline. No? And we can see here that we have a shallower oxygen minimum zone. This is the upper limit of the oxygen minimum zone, the depth of the upper limit of the oxygen minimum zone. And we can see that we have a shallower OMC since 80s until, for example, 2008. No? So that means that maybe we have a deoxygenation in front of Peru. So for this talk, we have two research questions. The first one is, what are the processes involved in the oxygen changes in the water column during ENSO phases, no? El Niño and La Niña events? And what are the processes that drive the oxygen trend during the last decade? Well, to answer this question, we used a couple model, no? This is the RONS model, that this is a physical model that give us temperature, salinity, currents, and 3D fields. And this is forcing by atmospheric forcing, no? For example, winds, head fluxes, freshwater fluxes, and also some of the input that needs this model is an open boundary condition that come from global models. This physical model is coupled to a biogeochemical model, PISCAS. PISCAS is able to reproduce, for example, the main cycle of the nutrients, for example, nitrate, silicate, and phosphate, sorry, and also can reproduce the dynamic of plankton like phytoplankton and zooplankton. And also we can reproduce the cycle of oxygen. Well, what is the configuration of the model? Well, we use 32 vertical level, and the horizontal resolution is 1/6 degree, that is around 20 kilometers. Well, it is an interannual simulation that spans from 1958 until 2008. We have each 6 date outputs. What about the forcing? No, we use a wind downscale from NCEP and head fluxes from COATS and NCEP plus NCEP. About the boundary condition that we use, well, The physical boundary condition come from the global model SODA, and the biochemical boundary condition come from CARS climatological data. We don't have able at that moment an internal simulation to use, no, like a boundary condition for biochemical part. And this is the domain of the model that that we run the model, no, that span in all the Humboldt current system. But we will focus only in the region, no, in front of the Peru. Well, first we show the evaluation of the model. No, this is the annual mean of the oxygen minimum zone depth. This is observation data. This is from IMARPE, and this is from CARS, and this is the model output. We can see here that we have a shallower oxygen minimum zone, no, than the oxygen minimum zone that we can observe of Peru in the part of in the ocean part, and the model is able to reproduce this structure, no, that we can observe in the in situ data. About the interannual variability, no, in blue line, we observe the data from IMARPE, and black line represent the outputs from the model. We can see here that we have a high interannual variability, but the model is able to reproduce, for example, the deepening of the oxygen minimum zone during El Niño period that is remarkable for these gray boxes. Well, this in this graphic, we show vertical sections, no, from observations and the model during neutral period, El Niño period, La Niña period, and the anomalies. We can see here that the model reproduces well, no, the structure that we observe, for example, of the oxygen minimum zone during the neutral period. We also have a reduce of the oxygen minimum zone during El Niño period, and also we have a showing of the oxygen minimum zone during La Niña for observation and model. And when we observe the anomalies, we can see here in this color represent that we have a positive anomalies. So this means that we have a oxygenation in this depth, no, for and is reproduced for observation and for the model, no, between more or less 50 and 200 meters depth. And during La Niña, we have negative anomalies. No, that means that we have less oxygen. Well, what are the process that involved, no, in the changes of oxygen during El Niño and La Niña? Well, first we need to talk about the Kelvin wave. No, the Kelvin wave are originate in the equator and cross all the equator and reach the South American part. No. And this propagate like a coastal trade wave, no, along the coast. But we can follow this coastal trade wave, no, follow the sea level anomaly, for example, no. When we have a warming Kelvin, warming coastal trade wave, we have positive anomalies of the sea level anomaly. And when we have a cold coastal trade wave, we have a negative anomalies. We can see here, for example, a Hoff-Muller during El Niño and during La Niña, that during El Niño we have the passage of two warming Kelvin wave, no? And during La Niña we have two important cold Kelvin wave. But one of the impact that we can observe here, this is the depth of the thermocline and this is the depth of the oxycline. We can see here that this passage of the Kelvin wave deepening the thermocline and also deepening the oxycline, no? So for that reason, we have more oxygenate water. The opposite we observe during La Niña, no? During La Niña, we have a shoaling of this cline. For that reason, we have less oxygen in this condition. Also, it's important to see, for example, a meridional section at 88 west, no? And you can see here, for example, the subsurface equatorial currents, no? We can differentiate here the jet and the primary and secondary jets in this red box. And in the black box, we have the equatorial undercurrents. Well, the direction is towards South America, no? And we can see here that during, for example, La Niña and El Niño, the structure of this current changes, no? When we compute the anomalies, we can observe that we have, for example, a decrease of the intensity of this current. But when we are in a warming period, we have a intensification of this current in front of Peru. So this also impact in the shape of the oxygen minimum zone. Also, we can talk about biological processes. No, we know that, for example, that we have processes that consume oxygen and processes that produce oxygen. No, between these processes, we can mention, for example, the remineralization, nitrification, the respiration. No. And during, for example, a warming period, we can observe here, this is anomaly, no, of a profile of anomaly of remineralization. That means that when we have a more negative value, we have more remineralization. So during a warming period, we have more remineralization due to we have more availability of oxygen. Well, the first part of this conclusion, no, of this first results is that in warm Kelvin wave deepens the oxycline. For that reason, we have more oxygenated water in front of Peru. During El Niño, increase also the concentration of oxygen, no, due to we have a intensification of the flux by jets, no, and also the consumption of oxygen, no, through remineralization increased due to subsurface oxygenation and in spite of reduced productivity during this event. Well, if you want to see more detail about this result, you can check, for example, this paper from 2019. Well, about the second part, about the trends of oxygen in front of Peru. This is the depth of the upper limit of the oxygen minimum zone. And we can see here, this is from IMARPE data in red, and in black is from the model. We can see here that the OMC, no, presents a shoalings. This is the shoaling since 70s. So this indicate maybe that we have a deoxygenation, no, in average. But when we observe, for example, this trend, but in all the region, no, we can observe that this deoxygenation is in all the region in front of Peru. Well, this trend also we observe in the column water, no? In blue color means that we have a strong deoxygenation, and this strong deoxygenation is related, for example, to the oxycline. When we observe the trends, no, a profile of trends of oxygen, we can see here data from IMARPE in red and data from the model in black. We can see here that a stronger deoxygenation that we observe is around 50 meter depth. And we love like 10 micromole per kilogram per decade in this at this level. So also we want to see a versus between remote forcing and the local forcing, no? Because I mentioned that this is important to the formation of the oxygen minimum zone. So we did some sensitive analysis. So in the first experiment that we did, we removed the interannual variability of the local forcing. So like the winds, for example, no? And when we don't have interannual variability of the local forcing, we can observe here, for example, in gray line is the interannual simulation that I showed before. And in red line is the result of this experiment, no? We can see here that we can reproduce a shoaling of the oxygen minimum zone, remove the winds, the interannual variability of the winds. But what happened when we remove the interannual variability of the remote forcing? We obtain a no significant trend of the oxygen minimum zone depth. So in conclusion, we can say here that the remote forcing drive the deoxygenation in front of Peru. But what happened in the remote forcing, no? So we observe, for example, here, no, what happened with the intensity of the currents, no, the equatorial undercurrents and the primary and secondary jets. And it's obvious that we have a decrease in the intensity and velocity of these currents, no, since 70s until 2008. So this influence and in that we have less oxygen flux from the equator. So we want to see if this is robust with all global model, and when we compare all the global model, several global models, sorry, we found that all reproduce a decrease in the intensity of the equatorial currents. So like conclusion of this second part, we can say that we have a shoaling of the oxycline since 70s until 2008. We have a decrease of the oxygen concentration, no, due to we have also a decrease in the zonal fluxes of the subsurface current, and the remote forcing is the main driver of the deoxygenation in front of Peru. If you want to see more in detail some result, you can check in Research Square that we submit a paper in scientific report. So it's available online, this result that I showed. So, well, that is all. Thank you.
Thank you, Dante, for your talk. And now we invite everybody to write their questions in the question box in order that Dante can make some responses. Okay, there is one question here by Julie Poulin. The question is, what do you think would be the impact of using higher resolution winds in your simulations? Would it capture, for instance, the coastal local oxygen minimums in the in situ data?
Okay, thank you for the question. Well, it's important to try to use high resolution winds because we see, for example, that when we use coarse winds, no, or with a low resolution, that dwelling is not well reproduced, no. So when we use a high resolution winds, we can reproduce, for example, better that dwelling. And I mentioned that that dwelling also impact in the oxygen concentration because influence in the that we have, for example, if we have more intense at welling, we have less oxygen in the surface water. No? So I think that it's really important to the modeling, try to use high resolution winds with high resolution.
Okay, thank you. There is another question by Benny Peter. Do you find any influence of mesoscale eddies or vertical flux?
Okay, well, I did not show in this presentation, but for example, if you read the paper about the impact of ENSO in the oxygen concentration of Peru that I put in the talk, you can see here that we have some analysis about mesoscale. During El Niño, we have more mesoscale activity. So that also influenced that we have more oxygenation in the upper part of the oxygen of the limit of the oxygen minimum zone. So also has also we can mention other, for example, paper like Soren of 2015, I think, that showed that the mesoscale activity, for example, oxygenate the upper part of the oxygen minimum zone. So it's really important, but well, we no focus in this presentation. Sorry for that.
Thank you. There is another question. This is written in Spanish, but I will translate it to English. It says, recent models of high resolution suggest a diminution of the variability of ENSO with future climate change. And the question is, how this could affect the deoxygenation in the Peruvian waters.
Okay. Well, in this case, in climate change, maybe will be the deoxygenation that we observe in future, maybe will be more related to have more intense mixed layer, a mixed layer, no? Because we don't have, for example, a change between subsurface water and surface water. But for example, a recent paper from Echevan, 2020, we can observe that the model, maybe not all the models reproduce a deoxygenation in front of Peru. So not all the models are agreeing that we have less oxygen in the future.
Okay, we have time for the last two questions. The first one is from Andy Dale. He asks, is the shoaling of the depth of the OMZ in the last decade statistically significant?
Sorry, I don't understand very well
He asked if the shoaling of the depth of the OMZ in the last decade was statistically significant.
In the last decade? Well, in the last decade, we observe, for example, because my analysis is until 2008, but in the last decade, we observe maybe a shift of the regimen of the depth of the oxycline, because some data, for example, from IMARPE, you can read the paper of Graco 2016, I think, showed that we have a oxygenation in Peru. And this is agreed, for example, with a paper from Atlantic Ocean, that we have a shift in the intensity of the currents. No? Maybe the currents in the last decade are more intense than in other periods. So this influence in that maybe in the last decade we have more oxygenated waters.
Okay, thank you. Well, it seems that we have the time over. I'm very sorry because there are many questions, which all of them are very interesting. And we invite you to contact directly to Dante in order to have a more dialogue about his findings. Thank you very much, Dante, and we move on to the next talk. Okay, well, we move to our next speaker today, Dr. Renato Salvatteci. Renato studied fisheries engineering in Lima, Peru. After a Master of Science degree in Marine Ecology in Mexico, he earned his PhD in Environmental Sciences at the University of Paris 6. From 2014 to 2019, he worked at the Institute of Geosciences, Kiel University, first as an Alexander von Humboldt postdoctoral fellow, and then as a postdoctoral researcher of another project. Since 2020, he's at the Center for Ocean and Society, working in the Humboldt Current Tipping Points project. Renato reconstructs past changes in the fish population variability at multiple time scales based on the innovative approach of using fish scales and bones deposited in marine laminated sediments. So now I say welcome again to Renato with his talk entitled Paleoceanographic Evidence for Smaller Fish Species in a Warm and Oxygen-Poor Ocean.
Thank you, Dimitri, for that.
Thank you
for the introduction, and thanks to the organizer also for inviting me to give this talk. Today, I will present some of my latest results that focus on the Humboldt current system. And well, I'm going to talk about fisheries, aquaculture, deoxygenation, and warming. So I will just give a very, very brief update of on the importance of fisheries and aquaculture. So first of all, food. Fisheries and aquaculture are important part of the food security, especially in developing countries. Global production reached 180 million tons in 2018, which is a lot. And what is important here is that it provides to 3.3 billion people with at least 20% of their animal protein supply. Another important thing here is regarding livelihoods. 60 million people are employed in the primary sector. And it's important also because the total engagement of women across both fisheries and aquaculture is just 14% in the primary sector. But when we go to the manufacturing, more than 50% women are included in aquaculture, mainly in manufacturing in developing countries. Trade is the other point on why it's important fishery and aquaculture. Value of world export of fish and fish products reached 164 billion in 2018. And the share of developing countries in total fishery export was about 50 or a little bit more than half of this quantity. So very briefly, the main areas of fishery in the world are the temperate areas like the North Atlantic, North Pacific, the tropical areas like the Ocean Pacific, and the upwelling areas. And here we can see the temporal trends in the catches in these three different areas. And the green line shows a declining trend since the mid-90s. It's mostly driven by a decrease in catches of two main species, anchovy and jack mackerel. And I'm going to talk about these species as well. And when we talk about upwelling areas, the most important are, well, the Humboldt Current, California, Canary, and Benguela systems. They are extremely important in terms of fishery production because they represent a very, very small proportion of the world ocean, but have historically provided around 20% of global fish catches. In particular, the Humboldt Current system is a key ecosystem in terms of fish productivity, where an extremely high biomass of anchovy has yielded almost 15% of the total global reported fish catch in some years. Well, it is clear that climate change will threaten some fishes and fisheries as well. So there are many stressors. And the first one is, I would say, it's the deoxygenation of the subsurface. I will also show this figure from Lotte Strama. So oxygen minimum zones are expanding in the tropical oceans. And we can see here in this time series of oxygen, where blue indicates waters with low oxygen contents. This figure shows that the oxygen minimum zone is expanding. So it's a time series from 1960 to 2008. And this is producing a compression of the habitat of some species that cannot tolerate low oxygen waters. Then we also know that the oceans are warming. Here we can see a map of the difference in temperature between 2015 and the start of the century, of the last century. And we see that most of the ocean is warming, with the exception of the North Atlantic, where the cooling is explained by melting of ices. And this can also cause changes in the distribution of some species. Climate change is also expected to change the timing and strength of wind-driven coastal upwelling. So the systems are changing. So one question that we have here is whether or not the eastern boundary upwelling system can help to feed 10 billion people by 2050. There's some hypotheses regarding the impact of warming on body size at different time scales. One thing that scientists know is that the climate change is expected to result in smaller fish size. And this could come from two different points of view. One is at the community level and the other one is at the population level. At the community level, there will be an expected decrease in mean body size, okay? But this could be produced either by a decrease in the mean body size of a specific species or due to an increase in the proportion of small species. Both hypotheses are not exclusive. And basically this shrinking of the body size has been linked to reduced oxygen availability and what it implies for metabolic scope for the fishes. What is important to learn here is that smaller fish tend to have greater capacity for maintaining high activity in warm oxygen poor water. So warm waters benefit the small in this case. The problem to test this hypothesis is the influence of fishery, because fishery takes the adults always from the fishery, and then it leaves, that theoretical link between the size and ocean warming and deoxygenation untested. And well, I'm going to talk about the Humboldt current system, so I have to give a very brief introduction about the main actors in the Humboldt current. And I want to tell you that oxygen plays a major role in the distribution of some species, or most of them. So here is a schematic representation of the Humboldt current with water, the cold coastal waters near the coast and subtropical waters offshore in red. We can also observe a representation of the oxygen minimum zone, this deep blue color. Well, and by the actions of winds, upwelling brings cool nutrient-rich waters from subsurface, promoting marine productivity in terms of phytoplankton and zooplankton. And anchovy in Peru is present near the coast, let's say in the first 30, 40 miles, in great abundance. And anchovy forages specifically or more on zooplankton than on phytoplankton, and can tolerate periods with low oxygen concentration, when the waters have low oxygen. Sardine, when it is available, which is another small pelagic fish, is located a little bit offshore in the front between the subtropical waters and the cold coastal waters. They need much more oxygen to thrive. There are also other species like mackerels and jack mackerels that they like more offshore and oxygenated waters. Also dolphin fishes, or it's also abundant of Peru. And what is really abundant of Peru is the biomass of mesopelagic fishes. There is a huge biomass of mesopelagic fishes of Peru. It's like 8 to 10 million tons. And these fishes are awesome because they have evolved physiological and morphological adaptation to live in oxygen deficient waters. They can tolerate episodes of severe hypoxia as well. There are other species, for example, during the last decades of Peru, we have observed a large biomass of giant squid. This species also is very well adapted to low oxygen concentrations. And there are other species like whale that they simply don't care much about oxygen. And well, most of these fishes, with the exception of the mesopelagic fishes, are currently targeted by artisanal and industrial fishery. And also we have a huge biomass of birds and for seals in the coast. So what are the projections for Peru for this century? They're not that nice, because the model projections, but always using high emissions scenarios, so it suggests that by the end of this century, the Humboldt current system will be warmer and less oxygenated than has been in modern time. So the future of the Humboldt current, but also of all the other eastern boundary upwelling system is uncertain. Here are two figures, one of temperature and one of oxygen. Temperature at the left, oxygen at the right. And this figure is just a difference of temperatures of the last decade of the 21st century minus the first decade of the 21st century. So we can observe a warming that goes, in this model, goes from up to 2 degrees in front of Peru, but there are other models showing up to a warming up to 4 degrees. And in the oxygen figure, we can see a deoxygenation in front of central Peru as well. So these observations raise an important question on how these environmental conditions will affect the abundance and the fish community composition in the Humboldt current system. So first of all, marines, I am a paleoceanographer, so marine sediment cores can reveal the state and dynamics of past climate and ecosystems. In particular, fishery independent data from sediment records offer the best opportunity, according to my view, to understand the response of the fish community to a warmer world. So here we can see a temperature record. It's an anomaly temperature record from in Antarctica for the last 400,000 years. Time goes from left to right. We can see periods with high temperature anomalies that we call interglacials, and periods with very low temperature or low temperature anomalies that we call glacial states. In this presentation, I will focus on the last 2 interglacials, the marine isotope stage 5E, which is highlighted there. It occurred 125,000 years ago. Temperature was much higher than the temperature during the Holocene. And well, and the whole Holocene, which is the current interglacial, is that the last 10,000 years. Okay, so very briefly, what are my paleo proxies? So I'm going to show you some results of my proxies or indicators. And I will focus on 2 paleothermometers. One is the Alkenon, which is a group of long-chain organic molecules produced by one type of algae. And also the TEX86, which is another paleothermometer based on the lipids of some archaea. Then I'm also going to show some N15 values, a record of N15 as a proxy for water column nitrogen loss, N2 loss processes, and the fish debris. So what I did was to identify the fish debris, and I also measured the vertebrate width to test for changes in the body size. So here is the first results. Here are 3 records of temperature inferred from alkenons for the last 140,000 years. Time is going from left to right. I highlighted the marine isotope stage 5E and also the Holocene. And the marine isotope stage 5E show temperatures up to 3 or 4 degrees higher than during the Holocene. Next, here are another 2 figures. The first one is the record of TEX86, and the other one is the difference between the alkenons and the TEX86. Alkenon temperature records the temperature, let's say, of the photic area of the ocean, the thermocline and upper thermocline, but the TEX86 record the temperature below the thermocline and below this area. So, and the difference of these 2, we interpret as changes in the, like a thermocline proxy, okay? Larger the temperature gradient during the marine isotope stage E than during the Holocene, it means that the thermocline was deeper. Finally, the last record is the record of N15. Well, we observed high values of N15 during the marine isotope stage 5E, which is consistent with an intensified water column denitrification during the last interglacial, as would be expected with a more intense oxygen minimum zone. So now with the fishes, now what I want to show here is this is the abundance of the different fish species during both interglacials. Blue for the Holocene, and then I will show you in red the fish abundance during the warm and oxygen poor marine isotope stage 5E. I will focus on some species, not all of them, so I will just put here anchovy, gobies, which is very important, Dinsingera lucetia, and blue lantern fish. These 2 are mesopelagic fishes. And in other species for the Holocene, we have a lot of other species like mackerels, jack mackerels, sardines, and hakes, but we are not going to focus on this. And I also inserted the inferred size of the fishes. You know, anchovies around 12 to 14 centimeters. The goby-like fishes that I found in Peru, just 3 or 4 centimeters. This is the size of the fish. The Vincingeria lucetia, 4 to 6 or 7 centimeters, and the blue lanternfish also very, very small, 5 centimeters. So what I found in the last interglacial was that the anchovy biomass was very, very low compared to the Holocene. The Holocene, anchovy dominated the Holocene, and I found a lot of gobies of Peru during the marine isotope stage 5E. A lot. The fish community was almost composed by gobies. And during the Holocene, I only observed 3 vertebrae, so almost nothing. And also there was an increase in other tropical mesopelagic fishes like the Vincingeria lucetia and the blue lanternfish. And in the case of the fish community in the last interglacial, the other species comprised a lot of other different mesopelagic fishes. So just to make a summary here, during the marine isotope stage 5E, no anchovies and a lot of small fishes like gobies and mesopelagic fishes. Now I want to show here the average vertebrate size of each species or group. Again, in blue, the Holocene, and in red, the marine isotope stage 5E. And in parentheses, I put just the number of vertebrae that I measured. When I combine all the species, you know, the Holocene show the largest vertebrae. And in my first slides, I talked you about this hypothesis on the reduction of the fish size. So it was either a decrease in the size of a species, you know, a decrease in body size, or an increase in small or an increase in species with shorter size. So regarding this body size shift hypothesis, I would expect a reduction of the vertebrate size during the marine isotope stage 5E. For example, anchovy, here both measurements were similar during both interglacial. So, and the same for Vincingeria and the same for blue lanternfish. So there wasn't a decrease in the size of this species, okay? So I couldn't find any evidence to support a decrease in the population body size. But what we found here is that we found smaller fish species. So this is the take-home message here. Smaller fish species and not smaller fishes from the same species flourished in the Humboldt Current system. So this suggests that there should be an environmentally triggered tipping point beyond which anchovy ceases to be a dominant part of the ecosystem and other species take over the system. So this is like a potential scenario in a warm and oxygen-poor ocean of Peru. Well, upwelling will continue to exist, you know, because some diatom data that I have, but I didn't show here, show that there was a normal upwell, like an upwelling like during the Holocene. Perhaps a little bit less productive due to a deeper thermocline. But what is important here is we also found more sulfidic conditions in surface sediments, and this may lead to increased frequency of sulfidic events in the water column, which is highly toxic for some species, but not for gobies, you know. And it seems that gobies will be the new king in the Humboldt Current system, displacing the anchovies. And this view of the Humboldt Current system, for me, is a reminiscence of the Northern Benguela Current system, where nowadays we have hypoxic conditions and the presence of H2S. And this resulted in the fishery collapse and the rise of a goby there, no? which has extreme tolerance to sulfidic events. Perhaps the biomass of some tropical mesopelagic fishes and also perhaps also that of giant squid will also probably increase because they are very well adapted to low oxygen conditions. And probably, you know, these other oceanic species like sardine, jack mackerel, and mackerel will have to move offshore or southwards, looking for more favorable conditions, and the same for anchovy. Now, the last slide of results here, I want to show a generalized additive model describing the significant relationship between the vertebrae width and the interaction between temperature and oxygen. Okay? So we have the size of the fish at the left, and at the bottom we have the temperature and the proxy for oxygenation. So I just want you to focus on two parts of the figure. The first one is a combination of less oxygen and low temperatures, which is at the back of the figure, you know, and it's associated with large fishes. And by large, I mean like anchovies. These are the conditions during the last century and some periods during the late Holocene. So that means that the strong oxygen minimum zone with relatively low temperatures were perfect for anchovy productivity. But if we look to the bluish area, of the figure, we see a combination of less oxygen and high temperatures. This bluish part again of the figure. This combination has never been observed in the historical record in the last 70 years, and it's associated with the smallest fishes. And this is specifically the period of the marine isotope stage 5E. So I'm finishing here, almost 20 minutes. These are the take-home messages. So fish debris deposited in laminated sediments indicate an increase in the proportion of small species in a warm and oxygen-poor ocean. So I didn't find any evidence for a shrinking in the size of a specific species. A warm and oxygen-poor ocean will not be good for anchovy, but other species will take the position of anchovies, very likely gobies and perhaps mesopelagic fishes. And what is important here, these fishes are extremely difficult to harvest. It's very, very hard to harvest gobies and mesopelagic fishes. And of course, they are less palatable than anchovies, and this will definitely indicate a threat to the global fish supply. Thank you.
Thank you very much, Renato. And now we go with the questions. The first question is by Emma Pontes. She asks, it sounds like environmental conditions are selecting for certain smaller species of fish. Could this idea be applied to other organisms' ecosystems? In other words, might we expect to see a shift in species compositions in other marine ecosystems?
So yes, and this idea that the temperature regulates the size of the marine animals, or any animals, come from the literature from the 50s or the 60s. It's not something new. Huh? And in marine ecosystems, there are a lot of papers, in fact, explaining these relationships between the temperatures and the size of fishes in general. Huh?
Okay, we go to next questions by Mark Costello. He asks, are the gobies benthic or pelagic? I think most species are benthic.
Yes. So most species are, that's a very good question. Yes, most species are benthic. Some gobies in the Humboldt, in the Benguela system, they are benthic, but they can swim in the water column of depth of, I don't know, 100 meters, 120 meters. So, but in Peru, I don't know what happened in the marine isotope stage 5E, but we have to take into account that there are some species that can change their benthic to pelagic lives. Huh? So yeah, it's the same with this red squat lobster in Peru, right? Now it's a pelagic and it's a benthic in other places.
Okay. Here, Mark Wisecki asks, could good or poor preservation of fish remains have generated a bias in fish abundance in your records?
Good question. Good question. The thing here is that many people will say, no, but this marine isotope stage is 125,000 years ago, so all your fish scales and fish vertebrae maybe are dissolved. But remember that the fish vertebrae that we found in the marine isotope stage 5E are the smallest, the thinnest one. So they are really, really thin. Okay, if there was degradation, I couldn't see any of them. Okay, so, and the preservation is very, very good. If I measure the preservation of fish debris, is even better than during the Holocene.
Okay, Elva Escobar asks, however, small mesopelagic fishes during the vertical daily migration is a current target of many fisheries to feed cattle or produce pellets for aquaculture. What is your point of view on this?
Yes, but remember that in Peru, we fish a lot of anchovies to produce fish meal and fish oil for aquaculture. 1/3 of the production of fish meal and fish oil comes from Peru. So if I'm saying here that the anchovy productivity will go to very low levels, you have to imagine all these, how this will cascade in all the other activities related to anchovy. Huh? All the fish production, all the aquaculture production, the salmon that we eat here in Europe, or it's the prawns, everything. It's all the aquaculture is, you need fish oil. So if we don't have this, and the mesopelagic fishes, they do not produce good fish oil. There are some papers explaining this as well. And some species of mesopelagic are even toxic for human consumption.
Thank you, Renato. We are moving with the last questions. Mike Roman asks, To what extent do you think warmer temperatures and lower oxygen has a direct physiological effect on fish versus lower oxygen and warming temperatures selecting smaller zooplankton species, which may favor smaller fish predators?
Yeah. Yeah, well, yeah, there's a point here because also in warm waters, we expect to see smaller zooplankton size, no? And this will benefit also the smaller fishes, right? Yeah, I have to think about this. Thanks for the question.
Thank you. Let me see if there are more questions. Okay, I have a issue here, Kirsten. I cannot see the last few questions. Maybe you can help me here. Okay. Okay, there is a question here. Are different oxygen requirements between anchovies and gobies? And the second half question is about availability of otoliths, if you were able to analyze them.
Unfortunately, in 15 years of counting fish there is in sediment records, I cannot find otoliths. The otoliths are, we think they are dissolved. I don't know, somehow they are dissolved because in other areas like Santa Barbara in California or in Mexico that I have been looking at the sediments, otoliths are abundant, but in Peru, they are not abundant. Maybe there is a problem perhaps of dissolution there. And about the oxygen, well, the gobies can stay in the oxygen minimum zone for hours. They have also developed some morphological strategies to cope with low oxygen. But anchovy, they can withstand time periods in waters with low oxygen, but not inside the oxygen minimum zone. They need to breathe. They need oxygen.
Okay, well. Thank you, Renato, and thank you to all attendees for your questions. There are many more, I know, and please feel free to contact Renato directly to have more insights on his research. Well, now, before saying goodbye, I want to announce the 53rd Ocean Leash Colloquium of the Global Ocean, which will be sponsored by the Global Ocean Oxygen Network. Low oxygen environments in marine and coastal waters. These will take place from 16th to 20th of May 2022. And the abstract submission deadline is in January 16th of 2022. You can see there the webpage of the colloquium, and we hope that we can see you all in this meeting. So with this, I thank you again for everybody, and we hope to see you in our next upcoming webinar. Thank you very much. Good morning, good afternoon, good evening.
Thank you.